So hello everyone, so welcome to this special public lecture. So this is the 7th Annual John Curtin Lecture in Medical Research, which will be presented this evening by Professor Carola Vinuesa. So my name is John Bekkers and I'm the Deputy Director of the John Curtin School, and I'm standing in for our Director who sends his apologies from his hospital bed, in fact. So before continuing, I would ask you to join me in acknowledging and celebrating the first Australians on whose traditional lands we meet, and to pay our respects to the leaders of the Ngunnawal people, past, present and emerging. So the John Curtin Lecture in Medical Research was set up a number of years ago now as a special way of honouring highly successful scientists in the school. And interestingly, this honour takes the form of a because we feel that outstanding researchers should also be outstanding communicators. Now, I don't think that this evening you'll be disappointed with the communicator that we have this evening, Carola. So, as I mentioned, this is the 7th John Curtin Lecture. 7 is a very nice number. There are 7 cardinal sins, and there are 7 wonders of the world, and Carola is one of the 7 wonders of immunology. So Carola was born in Spain. She did a medical degree in Madrid, and then she went to the UK to do clinical training. And while she was there, she, I gather, decided to move into research. So she did a PhD when she was in the United Kingdom. And then in the early 2000s, she moved out to the John Curtin School as a postdoctoral fellow. And in fact, I remember very clearly when Carola arrived, she was full of energy, as you can imagine. Now, Carola is one of our superstars. She's achieved a great deal of important work in the field of immunology, as you'll hear. So she came to the John Curtin School in the early 2000s as a young scientist, as a research fellow, and then she very quickly established herself as a very successful research scientist working on fundamental questions about how the immune system protects us against disease, and also in some cases how the immune system turns against us, causing autoimmune diseases. So more recently, Carola has turned to more clinical questions, and she'll be telling you probably a little bit about this more recent work, dealing with patients' diseases. Now, Carola has been awarded a number of very important prizes. So, for example, she was awarded the Science Minister's Prize for Life Sciences in 2008. She was awarded the Gottschalk Prize of the Australian Academy of Science in 2009, and in 2015 she was elected a Fellow of the Australian Academy of Science. So Carola is presently the Professor of Immunology at the ANU, and she's also the Director of the Centre for Personalised Immunology, the CPI, which is an NHMRC, a National Health and Medical Research Council Centre of Research Excellence. So Carola is a very fitting person to be awarded this special award, this John Curtin Lecture, and it's my pleasure now to ask her to come up and to receive the award certificate.
Thank you very much, John, for your I might just try this, given that I don't want to be leaning over for the entire talk. You can hear me, yes? Yes. Good. Okay, so I'm very grateful, John, for your very kind words, but also you for coming here. I think as a scientist, there's nothing that we like more than to be able to tell, you know, the public that we are trying very hard to solve some important questions, that things not always work out, but when they work out, it's very exciting, not just for us, but hopefully for people that can benefit from our discoveries, and that hopefully you can also pass a word to others that, you know, we use your money wisely. We know that it's the taxes that help us do what we do, and that we really try hard. Our teams work endlessly, you know, days and nights, to solve problems that we think are important. I'll try to give you a little bit of a feeling of what we do. I try not to make this talk very data heavy, because immunology in particular is a difficult field. But I also try to take you through the journey of how we're adapting to the new tools and technologies to try and make or discover and use all these new possibilities to discover what are the true causes of human, in my case, autoimmune disease. So thanks for being here, and I hope you can understand me. I know I have an accent. If you don't understand something that I say, I'm very happy to go through it a bit more slowly. So today we're going to, I don't know if it's going to work, because I'm going to have to be looking back, so I might try to have the computer the other way around, and let's see if this works. So I can try and, okay, I'm going to make things a bit worse. Okay, I'm sorry about this. This is not what we need just now.
That's all fine. Look. It's going to work just nicely.
Okay.
Here you go.
Thank you. Okay, so let's see if this works.
It should work.
So, perhaps if I do it like this. Yes, this is better. Okay. So my team has been working in the field of autoimmunity. And autoimmunity is a collection of diseases in which our own immune system, instead of focusing on destroying foreign invaders like microbes, bacteria, viruses, it turns against our own tissues and cells and destroys them. So some examples that you might be familiar with are, for example, type 1 diabetes, in which our immune system turns against the cells in the pancreas that produce insulin. And once they are destroyed, we don't know how to deal with sugar, and a lot of clinical complications arise. There is a long list of autoimmune diseases. I'll talk about one that we work a little bit more with in my laboratory, lupus, in a moment. But the fact is that there is over 80 of these diseases, and pretty much without exception, there is yet no cure for practically any of these. And this is really because we don't quite understand very well what are the real causes of disease, or what are the real molecules that go on to, trigger disease. So because we don't understand the actual causes, the molecules, the genes that cause it, we don't have very specific treatments. So what we tend to do is try and dampen the immune system with things like steroids or immune suppressants, some drugs that tend to be toxic, cause side effects, and very often they don't actually treat the most serious manifestations of disease. So we know that we need to understand better the molecular causes. I told you that in my lab we work in particularly with what we say is a prototypic systemic autoimmune disease, because it is a disease that targets many different organs and tissues. This is what we call SLE, or systemic lupus erythematosus. In this disease, there is a number of immunological problems. At the root of it is the production of rogue antibodies. Antibodies are proteins that are produced by B cells in the immune system that normally should be there to mainly fight viruses and bacteria, but they can actually start recognizing again our own tissues. They deposit in the kidney, they cause kidney damage. Sometimes they deposit between the different layers of the skin, and they cause rashes and other types of pathology. Eventually they can damage quite a number of organs. It goes with additional inflammation from other cells. But just for you to see how heterogeneous these diseases are, in order to diagnose lupus, a patient only needs to fulfill 4 of 11 diagnostic criteria. So here are the 11 type of criteria that patients can present with, and any 4 of these could constitute a diagnosis of lupus. 4 patients can present with a very different constellation of symptoms, or 2 patients can present with very different symptoms and both be labeled or diagnosed with a disease like lupus. And this one already tells us that probably there's quite a few different diseases in this big bag that we call lupus, and this is probably the case for quite a number of autoimmune diseases. So in order to treat these patients effectively, we need to know exactly what is wrong with each group of patients. So what we started doing, and this is very much what I started doing when I first arrived in the John Curtin, this was in the end of 2000, we took this mouse to human approach to try and understand the genes that cause autoimmunity, because that was what we could do at the time. And at the time, I joined the team of Chris Goodner, who was developing chemical screens in mice. So he was mutagenizing mice using a chemical that we call ENU. And then he was screening these mice for different types of immune disorders. At the time when I arrived, he was particularly interested in immune deficiency. So we realized that, so these screens, what they did is introduce random point mutations in the mouse genome, this treatment, and then you could screen for your disease of interest. So I started looking at autoimmune manifestations to see if we could find genes that could cause autoimmunity in mice. So in a way, this would mimic the natural human variation that we have in our DNA. So we would rapidly accelerate the introduction of mutations into mice. So I developed a series of screens at the time, and eventually we did have some quite interesting findings. We found genes that did cause something that looked like lupus in mice. This is one of the first discoveries. This is a mutation in a gene that was then unknown. We named the gene ROCKIN. We had a mouse that developed a syndrome that very much looked like lupus, with autoantibodies to nuclear antigens, big spleens, kidney damage. And this led us to also discover new cell types that were important for autoimmunity. Here we describe this particular cell that had a profile that looked like these cells that normally were localized to follicles, follicular T cells, which, doing some further investigation into these cells, we discovered some of the key molecules that have since been associated with not just these cells, but autoimmune disease. I won't give you all of the scientific information because it's a little bit overwhelming, but I will say that it really made us understand that it was the pathologic accumulation of some of these T follicular helper cells, or TFH cells, that was closely associated with lupus. In fact, only the mutant TFH cells accumulated in mice that had both healthy and mutant cells. And we found that there was a key molecule that appeared to be dysregulated that is called ICOS. Further, later on, we have discovered that ICOS is crucial for the development and the maintenance of these T follicular helper cells. And let me see what comes first. Okay, and then DU, who was a PhD student at the time in my lab, did some very elegant work sorting out the mechanism. How autoimmunity occurred in these mice was because the gene ROCKIN, when it was mutated, it couldn't regulate the RNA message for the gene. So a novel type of regulation at the time, instead of regulating proteins, it was regulating the first step before making the protein, which is making RNA from DNA. So there was too much of this molecule ICOS being produced, and eventually this caused this pathogenic accumulation of these blue cells here in the follicles that you don't see in the same numbers in healthy mice, that we know is linked to aberrant signals to B cells. Okay, so this led us to a few years of very intense and exciting discovery. We then realized that it was these TFH cells that were these specialized B cell helpers. We didn't know at the time which T cells were good for helping B cells, and B cells are the cells that make antibodies. So we gradually discovered quite a few important things that came with time. I mean, we and others, a few labs in the world contributed to this. So for example, the function of one of the most important cytokines that are produced by TFH cells is called interleukin-21. We now know that it's quite important for helping B cells in the germinal center, in structures where antibodies are made. We discovered the key factor that drives the formation of TFH cells. Again, this was discovered by DU at the time in my lab, and he published this a few years later than I've just added this onto this graph. But this was quite an important discovery, because when you discover the molecule that drives a cell subset, then you know that that cell subset is unique. So with all of this, I want to tell you that this basic research is important in that even though sometimes it doesn't tell us exactly how to cure disease, it really leads to a very significant amount of information that has been very useful for understanding not just autoimmunity, but now we know that these TFH cells play an important role, you know, in HIV, in transplantation. Both have protective and pathogenic roles depending on the disease we talk about, and we've learned how we regulate antibody responses. We then made on a few further discoveries. We discovered yet another subset in the germinal center that we call T follicular regulatory cells. So I told you TFH help B cells, TFR regulate, control, dampen antibody responses. So again, this was an exciting discovery because we could then see how we could manipulate this cell type to control the quality of the antibody response. We're still working with these CFR cells, and we think they are pretty important in the control of autoimmunity. So again, basic science that leads to discoveries of cells, of molecules that are important in the regulation of antibody responses and autoimmune disease. Another interesting, more recent discovery, unexpected players in antibody responses. In humans, believe it or not, a neurotransmitter, dopamine, we found was important for accelerating a good antibody response, a good protective antibody response. And we discovered a pathway that, again, I'm not going to bore you with the intricacies of the pathway, but it provides new insights as to how immune responses might be regulated. An interesting twist about this story is that in here, this mechanism or this pathway is unique to humans. We didn't find this dopamine regulated pathway in mice. So again, it highlights how it is important in parallel to do research with humans, with mice. Sorry, I don't know where I should be pointing. Okay, right. So then I've told you an approach that we used for many years, that we call the mouse to human approach to understand genes and pathways that contribute to immune disease. So not only random mouse mutagenesis has been useful, but there's been a lot of very useful information that we have gathered from what we call knockout mice that lack a particular gene or that we engineer to be deficient in a particular gene, or spontaneous mouse models of disease. But whether we like it or not, many of these approaches have still faced a significant block in translation, in that despite all of this knowledge for many years, we haven't really been able to develop better therapies. In the case of lupus, for example, the disease I work with, in the last 50 years, there has been only one new drug approved for lupus. And this drug still causes only very moderate efficacy. So what is the problem? We need to rethink how we're going on about this research. There's also been a large series of human efforts that we call genome-wide association studies that look for genetic variations that are found more common in a diseased population as opposed to a control cohort. And again, we have learned quite significant lessons about pathways and genes, but still we haven't been able to translate that very effectively. But we have learned some important lessons as well from these human studies. Some of these are listed here. Most common diseases segregate with common SNPs. SNP stands for single nucleotide polymorphism, so a single letter change in the genome. But in order to find these through these GWAS studies, they have to be common. In order to see them associating with disease, they have to be present in a significant fraction of the population. Because they are common, they don't tend to be very damaging. So there are changes in our genetic code that can be associated with disease, but they confer a very small increased risk for developing the disease. I might have to go to the mouse because this is going very slowly. Okay, so again, most of these genetic changes have very small effect on protein function. They can still point to disease mechanisms, but because, as I say, the risk that they confer for having disease is so small, they don't tend to be useful for individual patients in terms of changing their diagnosis or treatment. And even if you combine together all of this information, all of these genetic variants, they still don't explain the genetic heritability of disease. So there's something missing here, and how do we deal with it? I'm sorry, I'm going to have to go to the mouse because I can't really use the forward. Okay, can you hear me here? Can you hear me if I talk like this? Right. Okay, so what changed? Well, things that changed was the possibility of being able to look at individuals' genetic makeup. And here the initiative that really started with a big boost in funding by Barack Obama, started probably in 2015, is what we call precision medicine, that now has also received significant funding in the UK and now in the by the Australian government with the new Genomics Health Futures Mission. So what is precision medicine? Precision medicine, some people describe as seeing the zebra in all of us, finding what is unique to each individual despite all of us being related. And it actually focuses again in what makes us different. And one of the many definitions could be this one, is an approach to treatment and prevention that takes into account the individual's genetic makeup and also the unique environment and lifestyle of each person. And this really became possible because of having the draft of the human genome sequence. So this was really a critical moment in science for medical research. And it really transformed our understanding not just of the human genome, but of what we could do. And more than that, it was the technology that enabled us to then go and sequence human genomes. So in 2007, there was the invention of what we call massive parallel sequencing or next generation sequencing, in which it now became suddenly affordable and fast to sequence human genomes. So for example, the effort that took to sequence one human genome when it was first sequenced, it took about 15 years and several $1 billion. Now, thanks to these new technologies, we can sequence a genome in days, and it really now the price has come down to nearly below $1,000. So this is doable, and when patients need it, it really is. I'll show you how it could be economic to do this. And I will just emphasize again that these next generation technologies came about because of basic science. It was a basic science discovery, blue sky research, looking into new ways of, understanding DNA molecules that came with this discovery. So again, it's an emphasis that some of the most transformative discoveries in health come from fundamental research. So once the technology became available, and this is a graph that really only comes up to 2013, there was a huge exponential increase in the discovery of genetic mutations that cause disease. We found that rare and novel variants are present in individuals. They have arisen recently, and that's why they have avoided selection. They can explain previously unexplained causes of Mendelian disease. The novel mutations in particular, which are the ones that are present in an individual but not in the father or the mother, tend to be common causes of sporadic disease, a disease that only appears in one generation and you had never had it in the family. And there is now 6,000, some even say 8,000 of possible genetic causes of disease that most of these have been discovered in the last few years thanks to this technology. So we now can do either whole genome or whole exome sequencing. When we talk about whole exome sequencing, it's looking at the coding part of the genome, the part of the genome that makes proteins, which tends to be about 1% of the genome. This discovery of rare and novel variants, those might be unique to an individual or a family, is not possible doing this other type of association studies that I talked to you about a moment ago, GWAS. Now, we know that these novel variants are quite common. Each individual does carry, all of us, several, up to 100 of these variants that only you might have and not other members of the community. So with these possibilities, we decided to then change our approach. I told you that we started trying to do this mouse to human approach, investigate genetic causes in mice, and then see if we can translate them to humans. Now that it became possible to sequence human genomes, we decided, well, why don't we start with an individual and then try and see if we find genetic causes of disease, and more importantly, then try and understand how they cause disease. And to do that, we need to put them into cell lines or into mice, because we cannot experiment with humans. But very often it is also to just prove that those genetic mutations are really the causative ones. Because I told you a moment ago, we all have many novel genes in our genome, and for any disease you could possibly think that any one of 2 or 3 of these mutations could be causing the disease. So in order to be sure and to be able to change treatment, we need to prove that those particular mutations cause disease. So there's a number of ways of doing it, and I'll mention that in a moment. But then if we've introduced those mutations into cell lines or into mice, we can use those mouse models as well to test which drugs will be effective. Because now we will have the real mutations that cause disease in humans. Can we know then what are the treatments? In immunology, we are also very lucky because there's already a lot of drugs on the shelf that have been developed for many different purposes, targeting practically every known immunological cytokine or product. But at the moment, again, we don't know which patients will respond to what. So being able to test them in really well-validated or physiological models of disease is really precious. So we started this Center for Personalized Immunology thanks to funding from the NHMRC, and this is co-directed also by Matthew Cook, who has been a very long-term collaborator, and he is a practicing immunologist, a very good scientist, and we couldn't do this if it wasn't a collaboration with our clinical colleagues. And quite a number of investigators, some are here, these are chief investigators, but this is really a collaboration with a lot of clinicians throughout Australia and a lot of very dedicated professionals, students, postdocs. We have a website that you are very welcome to have a look at if you want to see in a little bit more detail what we do and the diseases we work with. Very simple, cpi.org.au. And what we really are about is about trying to understand what causes disease, so disease pathogenesis, understanding the genes and the pathways. And the ultimate aim is to improve diagnosis, to know exactly which diseases patients have, and to improve treatment, so that we really know what we can treat these patients with. So we deal with quite a lot of immune diseases, not just autoimmune diseases. Different teams in the center deal with different diseases according to their expertise. So this includes immune deficiency, for example, common variable immunodeficiency or primary antibody deficiency, a range of autoimmune disorders. Here I've just listed lupus, Sjogren's syndrome, rheumatoid arthritis, but we do look at quite a few of them, primary inflammatory diseases. And we take most patients through what we now have is a very established platform with different people that are specialized in each of these activities. So for example, we immunophenotype the patients very deeply, and I'll tell you an example of how we do that. We look at their genetic variants. We analyze these variants in isolation, as I say, in both cell lines and mice. We prove causation. We then reanalyze cohorts, and we test pathway-specific therapy. And again, this is a bit of an ideal pipeline. In some patients, we really don't get very far. In others, if we are lucky, we might get all the way across. So how we do that, for example, the immune phenotyping or the cellular endophenotyping, we want to know if there is any particular disease signature in that patient that might distinguish that disease from a different patient. So what we do is perform this technique that we call flow cytometry. We incubate blood cells from the patients with a range of antibodies. Now it's up to 40 different antibodies. And doing that, we can really map out over 50 different immune cell subsets that are in the blood. When you see this, you will understand why I didn't go into the details of the immune pathways that we work with, because they are quite complex. There's many different cells that come together and interact in various ways. But we now know that if we then try and look at the signatures, we get some patients clustering together with either B cell problems or immunoglobulin problems or CD4 T cell problems. So by doing this, we can start seeing that certain forms of disease associate with certain signatures, and that is useful, and I'll give you an example of why that is useful a little bit later on. We then, as I told you, take blood. Sometimes we can do it from saliva or just from a buccal swab, but blood tends to be a little bit better quality. We sequence the genome. We run it through a series of bioinformatic pipelines. Again, we are very fortunate here at the John Curtin because we've got really some of the best bioinformaticians in the country, I dare say. And this is not trivial, you know. When you analyze genome, a genome contains 3 billion letters. So to make sense of all of the variants in 3 billion letters, it's not trivial. But also the storage is not trivial either. We are again fortunate in the John Curtin. We have the largest supercomputer in the southern hemisphere. So we are storing genetic information, not just for patients in Canberra, but for patients in many parts of Australia and other parts of the world too. We've now developed a very user-friendly website, again, thanks to an incredible team based here at the ANU, that can bring in all of this genetic information together with the clinical information and some of the other details that we have from each patient, and helps us search through this in a very user-friendly way. We can very easily find out and sort the variants according to many different criteria. And these are some of the criteria that we use to try and identify which could be the variants causing disease. So we prioritize the variants according to those that change protein function. Of course, if a variant is de novo, it has a larger potential to be damaging. It has only appeared in that individual. It probably hasn't had time to be purified. We use a series of softwares that can predict how damaging a mutation could be in a protein. And we also try and use other databases that might have already found variants in that same gene in a number of diseases. So all of that eventually points to one particular change, perhaps in the genome, that we think could be causing the disease, but we are not sure. And if you're not sure, there's no way you're going to convince a clinician to change treatment, or it's not going to be useful for us to understand how that mutation causes disease. So again, we now can do something that was, again, unthinkable, you know, a few years ago. And this is, again, is thanks to another blue sky discovery that we call CRISPR. You might have heard about CRISPR because it has been in the news. Is a way of genome editing, of changing any letter in the genome with a relatively simple technique. Again, this knowledge comes from studying how bacteria defend themselves against viruses. So again, very basic research. And thanks to understanding that primitive immune system in bacteria, we now can use the same tools to very rapidly, very simply, very cheaply change any letter in the genome in either a cell line, or a mouse. And those changes can be transmitted then through the offspring. So we now can then investigate the mutant protein in isolation, either with biochemistry or through generating mice that have exactly the same mutation as the human. And this can be done in as little as weeks and cheaply. So what do? We've analyzed the variant with many biochemical and protein assays that I don't need to describe, but we can, if we have a cell line now expressing the variant, we can know how that protein works. Is it working better or worse? Is it now not able to interact with another protein partner? Is it causing something aberrant in the cell survival or proliferation? Sorry. As I said, we can make the mice, and with the mouse there is an unlimited things that we can do to understand how disease comes about. We can look at the cells and how they are behaving. We can try and identify signatures in the same way as we did with the humans. We can see how are these mice different in their immune system from healthy mice, and we might find just by doing just some very simple screens that they've got a particular subset of cells that is dysregulated. And that can point to a particular pathway. And with that knowledge, we could decide, well, actually, this drug probably might be effective, or we might have to develop a new drug. But having that is an incredible tool, because now we know that that mouse has the same mutation as the human has. So, coming back to the idea of signatures, you know, it's not going to be feasible to sequence every patient in the planet, but if we could identify signatures of disease that now we can link to a mutated gene. We could then go back to larger disease groups, entire cohorts, and instead of sequencing the genome, just look for that signature of disease. And we might think, well, even if it's not this particular mutation, it is related to a disease caused by gene X. So we pay quite a bit of focus in, once we've generated these strains of mice, to test them using a very large set of assays and find these biomarkers, these endophenotypes, these signatures of disease that could be a constellation of an accumulation of certain proteins in the blood or an expansion of a particular cell type. Because if we now know that the patient with mutation in gene X also has, let's say, an expanded TFH population, what we then need to go is screen a large group of patients with lupus for an expanded TFH population. And their disease might be related to this, the disease in these mouse. So the ultimate goal is to be able to stratify a large group of patients into smaller subgroups that share the same disease pathogenesis, the same molecular pathway, the same signature. So then we can start using these more specific treatments that we know work for that pathway and might not work for another one. So, great, okay. So we come back to lupus. How are we doing that? Well, we started applying this to these diseases and saying, well, we know that there is evidence already that there are single genes that can cause disease. In general, normally, most diseases are not caused by a single gene, but if you look at the most extreme phenotypes, at the severe end of the spectrum, at childhood onset disease in general, you might find the most severe forms that are caused by a very catastrophic mutation in one gene. And the list of these monogenic variants is increasing, so we asked, well, can we find the spectrum of genes and mutations that cause a disease like lupus? And you could argue, well, most patients won't be these type of catastrophic forms. Is it really useful if you really want to understand a complex disease? And we make the argument that it is, because even if only a small fraction of patients have these rare variants, they might point to a critical pathway that explains disease. And a very nice example comes from cardiovascular disease, the discovery of rare mutations in the LDL receptor. So this is a receptor for what we call the bad type of cholesterol. So even though patients with cardiovascular disease, only 1 in 500 people might have these very rare mutations, the discovery identified controlling cholesterol as a very important way of lowering the risk of cardiovascular disease. And now cholesterol lowering agents are given to most patients that have risk factors, regardless of whether they actually have the mutation. So again, finding these damaging mutations can help understand the disease, and finding drugs that will be effective in a subset of patients or perhaps in a larger group of patients. So these are some examples of cases, and I'm not going to give you many because many of them are actually quite difficult to sort, and we are still dealing with mouse models and trying to understand pathways. But this was one of the first ones that we identified, and this work was done by a very brilliant postdoc in the lab, Julia Elliard. And what we had is a very small girl that presented, 2.5 years old, with already the diagnosis of lupus at that very early age. So we thought these are the cases where we could find these very catastrophic mutations. When she was only 4, she developed a brain infarct, stroke. Again, very catastrophic manifestation, and it was diagnosed as cerebral lupus. So lupus patients do sometimes have these very unfortunate neurological complications. So sequencing the genome of this entire family revealed that the patient, and only the patient, had 2 copies of a mutated gene that is called TREX1. At the time, it hadn't been associated with pediatric lupus. Just one heterozygous mutation, a single copy, had been associated with a form of adult lupus called chilblain lupus. So what Julia was able to do is test the mutant protein in the laboratory and show that it was less capable of degrading DNA. And we found that what was the problem is that, so if you don't degrade DNA, you accumulate DNA in the cells, and this elicits a type 1 interferon response. This is a response that we normally make when we have viral infections or some types of infections. And normally it is good to alert the immune system that there is an infection, but if you do it in excess, if you produce an excessive amount of this type 1 interferon, you can start causing inflammation. And still we don't completely understand if this is the only cause of lupus in this type of patients. We are trying to understand if it's only the type 1 interferon, but clearly there are contributors. And you can see here that in this particular patient, the one on the left, but not the parents, they have a very striking what we call interferon 1 signature compared with, this is just a control of cells where we've elicited this response. So by knowing this pathway, you could say, well, this girl could be a candidate for treatment with a drug that blocks type 1 interferons, which actually is at the moment in clinical trials. So it starts pointing to therapies that might be effective in some patients. And now we have a mouse model that has a mutation in TREX1, and we can start to use that mouse model to try and understand how disease develops. Is it just type 1 interferon? And Julia has some very exciting data suggesting that actually type 1 interferon might be important, but it might not be the only thing. So by understanding this, we could refine even more the treatment. This is an example of how doing sequencing, it can actually help in the diagnosis of very rare disorders. So here we had referred to as a case of another very young girl with lupus. And we had been told that she had some unusual manifest clinical features as well, some hearing loss, developmental delay, and some unusual facial and limb features. And then she had some seizures, and only at the age of 17 she was diagnosed with systemic lupus with severe nephritis. And it was by doing the exome sequencing that what we found was that this girl had a syndrome called Kabuki syndrome that hadn't been diagnosed by the clinicians because they hadn't quite recognized all the clinical manifestations. But by knowing the mutation, which was a deletion that changed the protein of, in this case, KMT2D, which is known to cause Kabuki syndrome, this caused a loss of the protein. This girl could be diagnosed with a syndrome that has been frequently associated with autoimmune disease, although this was the first association with lupus. And again, it might not be the only genetic cause in the girl, but probably is a contributor. So genetic sequencing can reveal quite a few surprises, can refine the diagnosis, can help put patients on the right path, or at least I try to identify better therapies. I'm also going to tell you some interesting findings that we have where genetics really proves to be very surprising and can challenge our assumptions of what, in this case, probabilistics look like. This is a family that we had referred from Macedonia. I'll show you now that we have a very international network of patients that presented with a very unusual immune constellation of symptoms, including inflammation, infection. It was very sad when we were first referred this family. 3 of these children had died, all of them in the early years, months of life. So as you can see, one died at 20 days, 2 months, 3 months. The 4th twin was alive. And we did exome sequencing, and we found what was a mutation that has only very recently been linked to this type of immune disease. And it was very unfortunate because all of these infants were homozygous, had 2 copies of the mutated gene. The parents, each of them had 1 copy. Now the chance that you are homozygous in each child is 1 in 4. So these children had, all of them, this very 1 in 4 chance. But it doesn't end there. You will notice that the 4 are males. And what was pretty unusual is we actually went on to find yet another mutation that is X-linked, is only present in the X chromosome, so it only causes a disease when males have it mutated. And it actually acts in the same pathway as the first mutation. And we've been talking to experts, we've been doing some analysis, and it looks like probably both contribute to disease. So what are now the chances that the 4 have 2 copies of a mutated gene, and then a second mutated gene that only affects males, and the 4 are males? So if you calculate the probabilities, 1 in 64,000, which if you go by probabilistics, most people would have said, look, that's impossible, before we had genetics at hand. But now we know that when it happens to you, the probability could be 100%, no? When it happens. So for us, this is very interesting because there's quite a lot of lessons that we've learned from this. So extended family, sorry, this is, and as you can see, these are the only 4 individuals that have this fatal combination of these 2 genes and these mutations. Now, why? The sad thing, the other thing that I will tell you was very beautiful is, well, very beautiful. It is quite sad now, but when they referred to us this 4th patient that was alive, we had just made a mouse model for this, for the first genetic mutation. So Ansa Menders in our team was working on that mutation because they had had a completely independent family through another collaboration that he has with a mutation in this first gene. And we had made a mouse model with CRISPR that had that type of immune disease. And through the clinicians, what they wanted to know is there anything we can do for this child? Well, thanks to the mouse, we knew that the mutation, the defect, could be cured with a bone marrow transplant. At least in mice, it could be. So we gave the advice to the clinicians, you could try a bone marrow transplant. Sadly, well, in Macedonia, they couldn't do it. They had to refer the child to Turkey. The child traveled to Istanbul with the parents, and the day before the transplant, the child passed away, which was very sad, but it tells you that in an ideal world, in some other case, things could have, perhaps helped. But there are other things that can help the family. We know that by knowing this genetic information, the parents who are young, they are 23 and 25-year-old, they could still have a healthy child if they now have some type of prenatal diagnosis or pre-implantation genetic diagnosis. And thanks to Todor in our center, who is very proactive, he went to the Macedonian government and managed to get them free PGD, which in most countries is quite expensive. So there are things that could change the lives of patients. Now, as I told you, I've been talking about this part of the spectrum where we have Mendelian disease, one single mutation that has a very dramatic effect. As we move to the more complex adult onset disease, we think that in many cases it won't be a single mutation. There will be various genes that will contribute. And this is the challenge that we are facing now in genetics. How do we move to understanding the more complex part of disease? Still, we do find that some patients sit here with still 1, 2, 3 genes with very strong effects rather than many with very weak effects. But this is what we are facing now. And we've been doing some interesting research here. We have a few interesting leads, but I don't want to bore you with all of this. I will just mention that thanks to the Center for Personalized Immunology, we were able to launch, and this is really the merit of Matthew Cook, what is now called Canberra Clinical Genomics. So this is a service for the Canberra community to be able to access genomic medicine when the physician believes there will be a gain from sequencing the genome. So patients from different areas in medicine, particularly rare pediatric disease, but also there's been now cases in neurology, in cardiovascular disease, cancer, that have benefited from this. So we had a very significant budget from the ACT government, and the idea is that these patients have exome sequencing. If there is an immediate diagnosis, that is reported to the clinician. If there isn't, they enter into the discovery pipeline so we can do some research and try to understand what's going on. And there's been quite a few nice stories coming out of that. There's been already about 70 patients recruited and about half of those diagnosed. We also now have a center in China with a very significant budget from the Chinese government and a very beautiful team of people that are very talented. Martin is here. Martin is a joint PhD from both centers. And as I say, UK and Chiang Mai, for example, were here the week before last. For us, this is Very important because, you know, China has a very significant incidence of lupus, but also a very large population, and lupus in China is more severe. The end organs are more severely affected. And, you know, if we want to solve rare diseases, or it's very important to have more than one family with one mutation. So by joining forces and being able to do this collaborative work, I think we are both benefiting from this from this collaboration. So I would also say that we now also have a network of clinicians in other parts of the world, particularly in Eastern Europe, Turkey and Oman, who are asking us for help sequencing patients, but of course we are benefiting from being able to do better discovery exercises, as I say, when we have more of these rare mutations that we can use to understand how disease comes about. And just to end, what are the outcomes of precision medicine? Well, we are changing classification of disease by finding that, you know, some diseases are not just a single disease, there are many. We are not focusing so much on phenotypes, on organs affected, but on pathways. That refines diagnosis for each individual patient. We can start thinking of more targeted therapies. And what I think is very important is we can start stratifying patients for clinical trials. Again, we are early days, but you know, there's very few drugs that pass clinical trials, and this is because, again, they are tested against a very heterogeneous group of patients. If we can now select those patients that have particular pathways or genetic variations, it is more likely that they will start seeing much better responses to some of these drugs. And what are the challenges for precision medicine? Well, as you know, there are still quite a lot of concerns, particularly in Australia, about data privacy. You know, we haven't sorted what are, you know, health insurance, what are going to be the policies there. We now know that there is very active interest by patients groups. They sometimes are sequencing their own genomes. They are going to the doctor and wanting to participate in these studies, and I think that's very good. In fact, the challenge, probably the success from precision medicine will come from a true collaboration between patients and clinicians. There is a huge problem in working with all these large data sets. We need to look at, I mean, the larger the data sets and the more we share them, the higher the chance that we will be able to determine very accurately the frequency of some of these mutations and find different cases that have the same type of genetic lesion. But we have to optimize the collection of all this data. We have to find ways of dealing with the data. We also have to get better at managing the patient's genomic data, you know, integrity, long-term use. Can we reanalyze genomes from patients regularly? How do we incorporate that into our medical records? And then there is issues of educating the medical workforce, because genomic medicine is happening now, and we still have generations of, you know, clinicians that haven't been trained in this type of work. And there are cost versus efficiency gains. We all know that the immediate cost, if we decided to sequence all of our sick patients, would be substantial, but we know that in the short to medium term, there will be significant gains. And where are these gains going to come from? There will be less waste of drugs that are expensive, because at the moment we are just trialing drug after drug in patients to see which will work. We might not need to do that. And we will rely on less expensive diagnostic modalities. We might think sequencing a genome is expensive, but if you compare it with 10 different investigations, you know, MRIs and PET scans and a lot of tests that many patients that we really don't know what's going on with them go through over their lifetime, this one single expensive test might actually not turn out to be expensive at all. Plus the time to diagnosis could be shortened very significantly with a lot of, you know, savings that you can imagine. So with all of this, I have quite a lot of acknowledgement slides, because of course there's lots of different teams working in this. These are the main investigators in the CPI project, but of course I've got a team of very dedicated PhDs, postdocs that do a lot of the hard basic science, not just the applied science work that I tell you is enormously important to really come to understand disease pathogenesis. And probably we don't have time to go through each of these, but you know, this is why things work, because you have people working, you know, day and night And some of them are here even though they are submitting the thesis this week. So that is really a supportive team in every type of way. And of course, I've mentioned the China team, and we hope this grows. Exceptional researchers and a lot of collaborators. This couldn't be done without, as I said, lots of people working and generously providing time, effort, patience, etc. Quite a few different sources of funding. And with that, I want to end. Thank you very much.